about our company and about who we are and about this upcoming program that we will be starting next week. Okay, please do let me know if you can't see my screen or you can't hear me well. I'll make sure that I can pause and restart if that's needed. So the Omics Logic program for genomic data analysis will be starting next week. And we're excited about this program that has been piloted in several universities already around the world in the US. We've worked with a lot of faculty and groups, including primarily the Tauber Bioinformatics Research Center, which is a research center out of University of Haifa in Israel, where the team has been working for the past decade to develop cutting-edge computational tools for genomic and multi-omic data analysis. So we're excited to now make this program available online, and we'll talk a little bit more about the topics that we'll be covering, who is going to be involved, and how this is practically going to work, hoping that everybody is familiar with the topics and we can start our program next week. So what we want to do in this program is to bring awareness about genomic data, types of analysis techniques, interpretation techniques. And this is going to be a very comprehensive program that will be exclusively focused on the genomic aspect. And we'll talk a little bit about what that actually means in the context of a variety of projects. And we will have 16 online meetings. It's going to last for about 3 months. And then we will also have extensive time with support from our staff that could learn in the context of an independent project or a project that we work on as a group. And what we are going to do throughout this program is bring some of our team members who are going to be covering specific topics, supporting the research projects, or helping identify data sets and ways to analyze the data for each participant of this program. What we want to do today is kind of talk a little bit about, what are the goals of this program? What are some of the resources that will be available during this program? How will this actually work? And then we'll show you some of the things that we've prepared and also introduce you to some of our team members at the end. So as you all know, DNA, 3 billion base pairs, we have the human DNA. So a lot of information in the DNA code that basically refers to the variants in those nucleotides or groups of nucleotides that you can analyze with a variety of statistical methods, machine learning methods, but also interpretation is important here because in so much information, finding the information that is related to a specific phenotype or that could be informative in an understanding of a biological problem is the main challenge. So what we'll do in the program is, first of all, go through some of these introductory things to refresh everybody's memory and understand what are we dealing with. When we say genomic, how is genomic different from any other kind of omic? What are some of the technologies that are used to sequence this kind of data? What are some of the analysis methods? And what are some of the challenges that we are trying to solve when we analyze, process the data, annotate the data, and try to interpret this data in the context of a biological question? Then we will talk about specifically processing of genomic data. How do we address the different types of sequencing, for example, whole exome sequencing, whole genome sequencing? What are some of the attributes of next generation sequencing that we need to be aware of, like sequencing depth? How do we map the different mappers that are available? What kinds of references do we use? So we will really go step by step into all of those aspects, and we'll actually do this in a number of meetings. What we've found is that over the course of the past couple of years, working with a variety of groups that included groups of biology and clinical researchers, as well as students, starting from high school and bachelor level, we've found that it's very important for us to make sure that we build it up, the information, to the level of everybody's understanding so that everybody can contribute to the actual analysis and interpretation of a data set. And so we've been doing this for quite a while. And we structured this program the way that we think is going to be effective for anyone, irrelevant of their background, to be able to go from knowing something about genomic data and genomic data analysis to actually being able to do it in the context of their own question, their own biological question. So, for example, some of the things that we think are important here is the ability to really understand the various next-generation sequencing techniques that allow us to study a variety of biological, molecular biology phenomena. Like, for example, we can look at variants in DNA, we can look at expression of RNA, we can look at DNA methylation, we can look at microRNA signaling, and other aspects of what's happening at the subcellular level. But understanding what type of data and finding the appropriate methods is critical for us to be able to analyze that and get meaningful insights from the data. We also understand that not everybody is technically equipped to process that data independently. So we've created an environment where you can both learn and do the practical activities anywhere without having high-performance computing computational environment, without having the technical coding skills, and without really knowing the ins and outs of the methods. So we will try to get quickly to a practical, hands-on session where everybody gets to analyze data and start looking at what is inside a data set. And so the first step is really understanding what is this data. Then we'll talk about what kinds of variants, what kinds of DNA variants are we expecting to find in this data. Because we're studying the DNA, we are interested in a variety of mutations. For example, germline mutations in the DNA that are passed on from generation to generation. They're hereditary. Or somatic mutations, mutations that appear as a result of cell replication, and often occur as a result of some irregularities in the mechanisms that control cell replication. So somatic mutations, hallmark of a variety of cancers. We'll also talk about non-point variations, for example, copy number variation, chromosomal instability, and others that are an important aspect and require a specific type of an approach that is different from the analysis of germline or somatic mutations. So we'll dive deep into that, and we'll talk about how DNA varies, what do we use to analyze that kind of data, and then how do we actually use that information. And we'll have a couple of sessions just focused on some of those aspects. So we'll see how there's a number of different types of, for example, single nucleotide variations, substitutions, insertions, deletions, or as we commonly refer to them, indels. And how are they different from single point mutations in terms of their impact on the product of that specific sequence, oftentimes thinking of it as a way to understand the data, the raw data that we find in the sequencing reads, and try to align it with other information that we can gather from a lot of different other databases to understand the impact of those mutations on the overall sequence. We'll also talk about the analysis of these mutations. Once we have identified all of the positions where there is a change, the question is, what are those changes going to do? What is going to be the impact? For example, the impact on a gene. A lot of times we focus on, for example, whole exome sequencing, where only the expressed genes are analyzed for variants, to try and understand how those variants would impact the codon sequence, which will have an impact on the protein that is being produced from the DNA via transcription to the RNA and translation into protein. And so we'll look at the relationship between the sequence of the amino acids and the sequence on the DNA. We'll also look at different types of mutations, missense, nonsense, frameshift mutations, mutations that have a different type of impact, And we'll look at other methods, in silico methods, that will help us understand the impact of mutations and how significant they are. Also, it's important to annotate those mutations, annotate those regions where we found a lot of mutations, or even single nucleotide mutations, using reference databases or other types of studies that produce a lot of reliable, consistent results, as well as looking at other statistical methods that help us correlate genotype to phenotype. So we'll take a look at several examples where we can use the information in the DNA sequence as a result of processing that data and then correlating it to a specific phenotype, like, for example, arising mutations that come as a result of treatment, or that help us find the differences between genomes of infectious agents, pathogens in different regions. We'll also look at the protein structure impact and the function, and we'll talk about what are some methods to understand population genetics. How do we look at phylogeny? How do we understand the relationships that are defined by the sequence in the DNA? We'll look at some important studies that have been used or that have been actually developed to find enough consistent signal in a specific variant, in a specific gene. And one of them, of course, is TCGA, the Cancer Genome Atlas. The Cancer Genome Atlas is a study of thousands of patients across a variety of cancers that allows us to look at an individual gene level, individual nucleotide level, as well as associate phenotypes with some of the genotypes and other types of omics data. So the advantage is also that they have multiple levels of omics data, and we'll touch on that in the context of trying to interpret the functional outcome of a mutation signature. We'll also look at some of the other resources that are available, some databases that were created specifically to annotate a variety of mutations in different contexts. In the context of cancer, where we're talking about somatic mutations, in the context of germline mutations, in the context of different types of genomes. Then we'll look at machine learning for genomic data. So machine learning has become a hot word, a buzzword that everybody uses in a variety of contexts. But when we talk about machine learning and we talk about these different analysis techniques, it's important to remember what goes into the preparation of data for those methods to use the data that we have. Also, at the way that they work, and they work in a variety of ways because there are several groups of methods that are going to be applicable to this kind of data. And then we'll look at the outcome. How can this practically be used in a variety of contexts? Where is the benefit from having a data-driven model, like a machine learning model, that can, for example, predict a certain outcome of a mutational profile in a variety of samples that we feed into that model? So we'll look at the predictive aspect, we'll look at the data mining aspect, we'll look at how we can use different statistical techniques like regression and understand the factors and the features of our data. We'll take a look at the supervised machine learning models where we can look at classification, where we can look at discriminant analysis. We'll look also at the outcome, the mutational profiling, and how we can develop a signature or a biomarker using DNA data. Now, we'll spend a lot of time on the machine learning aspect because in a variety of settings, depending on the data type that we have and depending on the problem that we're trying to solve, we need to understand where to apply the different techniques and also how to combine those techniques together to help overcome some of the limitations of these conventional machine learning tools. We will look at the variety of methods and how they work together and where their application gives us an advantage when we look at the DNA data. And we'll specifically focus on preparing that DNA data. For example, when we have single nucleotide, example, they include information of change from one single nucleotide letter to a different letter. They also have a significance, and then we can organize them in a variety of ways based on their position on the DNA. So when we talk about a variant, it's important to understand how many characteristics or descriptors we have of those mutations to try and prepare the data in the appropriate way. But we'll look at the application of, for example, unsupervised machine learning methods like principal component analysis, clustering, network analysis, and we'll look at those. How can they help understand the variability of samples, how they can group those samples in a variety of ways, and help us mine the data for some patterns? How can we find patterns that are related between different samples or groups of samples? Then we'll also look at supervised machine learning, where we will use regression as a way to estimate relationship between some factors, and then understand how to train a model so that we can have a predictive output. And that falls under supervised machine learning, where we can have classification, regression, discriminant analysis. So we'll look at some of the applications of those methods for genomic data. And we'll do this in the context of several examples, and those examples are going to help us understand the biological significance as well as the limitation of some of these methods and the type of data that would have to be produced if we would like to use some of the methods that we are hearing about. Now, that brings us to an important part of this program. The program is structured to provide some basic information to give us a background so that the biology can more easily be related to the data. It will also give us an overview of data types, and there's a variety of data types that we need to consider in the context of genomics. Then we will also talk about the different methods and the steps to go from raw data to then processed data that we can, for example, structure and look at and visualize, to then analyze data, where the data went through several transformations and now, for example, could be compared between a variety of samples in a categorical or a non-categorical way, where we can look at either mutations or abundance of mutations in a particular region. Then we will talk about how do these different pathways and pipelines apply to a variety of challenges, a variety of project challenges. Now, we source these projects from public repositories, so the full data set is available, and typically these are impactful studies that come from high-impact journals. And we want to, first of all, understand how did people that led these impactful projects, how did they use the same tools, and what kinds of conclusions did they arrive at with the tools that they used. Using the same tools, we can then start thinking about our own questions that we might want to develop in our own project. So what we'll do is we'll look at several examples. For example, in cancer, how can we understand the significance of cancer for classification of different types of cancer or for a difference between a variety of cancers? How can we look at development of treatment resistance to, for example, tuberculosis and the resistance to the different types of treatments? Knowing the treatments from around the world, able to collect data from around the world in a consistent manner, can help us differentiate between the types of resistant and non-resistant strains, identify what the resistant mechanism might be, and also apply the same information to new samples so that we can understand what that. We'll also talk about case control studies and genome-wide association studies. So we'll talk about how do we use this kind of information to find global associations between, for example, a phenotype, and this could be related to a human disease or it could be related to something completely different. For example, rhizobacteria. How can we analyze genomic data from rhizobacteria, which is bacteria that grows around the roots of certain plants, and understand the impact of that mutational profiling on a phenotype, for example, crop production. But we'll also want to demonstrate how different techniques vary between model organisms, non-model organisms, organisms that have a defined protein and gene structure, and those that have a lot of overlap, like in the pathogen world. We'll look at a variety of environmental factors that shape the responses that an organism can pose to those environmental factors. So we'll talk about mutational fitness, for example, in pathogens. We'll talk about drug resistance as a response to an environmental pressure. We'll talk about the way that this type of information can be useful for cancer studies, where, of course, we are looking at a variety of factors: ethnicity, different ages, different locations, different diets, different backgrounds, different hereditary information, and different treatments. So being able to leverage the right method, apply it to the right data set, and understand the question in the context of data and methods is really the objective of this program. Now, here are some of the practical examples that I've referenced. In cancer, we'll look at somatic mutations, signatures of mutations, and we'll look at copy number variation and other non-single nucleotide mutations that are going to be important for the understanding of factors driving hereditary risk and staging of cancer, as well as differentiation of cancer. We'll look at tuberculosis as a way to understand drug resistance. We'll look at Ebola as, you know, the epidemic that happened between 2014 and 2016 gave us an insight into the way, because of next generation sequencing, we now were able to, first of all, understand the variety of genomes that we all call Ebola virus as a whole, but in reality, these are substrains, and how we can categorize that information to understand maybe which mutational profile contributed to virulence, contributed to pathogenesis, and also maybe the severity of the outcome. We will also use that information to try and apply that to our data sets where we can find, for example, a different landscape of mutational profiles, for example, in the new outbreak that is going on right now, or some other outbreaks that have happened in the past. And as I mentioned, rhizobacteria. So it's important to understand that the DNA code is a source of rich information about a variety of organisms and a variety of problems. And so we wanted to give a wide overview of some of those ideas. Now, before I jump into the speakers, I want to say that the project examples that we prepared are really built to give you some practical hands-on experience, to give you an understanding of the biological problem, but also to give you, and the program is structured to provide sufficient time for each one of you to be able to submit a project idea that you think these types of analytical methods could be applicable to. And then we will support your research effort in that area and do whatever is possible in the time that we have together to get it to a meaningful outcome in the context of the program. Now, I want to introduce you to our team and maybe let them say a few words about them. And first, I would like to introduce Despina, who is now working on preparing the sessions, the materials, and the project examples. So Despina, maybe you can say a few words about yourself.
Hello, everyone. I am very happy that we will be doing this course together for the next months. I am a bioinformatician. I am from Greece. I have a background in computer science, and then I have a degree from the University of Athens, and then I continued my studies in bioinformatics, again at the University of Athens. Then I worked in a company as a programmer, nothing to do with bioinformatics, but right now, for the past 3 years, I am a bioinformatician in a molecular diagnostics laboratory in Demokritos. It's the biggest research center in Greece. So in the lab, I am the only bioinformatician. I was the only bioinformatician up until 4 months ago. Everyone else is a geneticist, and we study hereditary cancer. We do targeted panel sequencing. We study genes that we know, or we have a suspicion that they predispose to cancer syndromes. And I am a PhD candidate as well. In my PhD, I developed some algorithms and pipelines for the analysis of DNA data from next generation sequencing. So the goal of our research in our lab is to decipher the mutational spectrum of hereditary cancer in Greece. It is so in Greece we have many subpopulations that are a little isolated. For example, in villages in mountains or in Greek islands. So we have mutations that are only found there and are in a. Is someone talking? I can't hear you. I'm sorry. Okay, so if you want to tell me something, because I can't hear you, I'm so sorry, you can write in chat. It's okay. So what I was telling is that, okay, so we study the population, the subpopulation isolates from Greece, and we have found many founder mutations that are found only here in Greek people. We are members of large scientific consortia such as SIMBA and Enigma that are that study hereditary cancer and specific cancer syndromes. And for the past 10 months, we are one of the 4 precision medicine units in Greece. There is we are trying to build a Hellenic precision medicine network here in Greece. Right now, we are trying to set this up. So in our lab, we are studying hereditary cancer for this precision medicine network. The goal is to provide cancer patients with genetic testing so they can benefit from targeted therapies. And I am responsible for everything related to bioinformatics in hereditary cancer for the network. This is more or less. I would gladly answer some questions if you have any.
Thank you, Despina. Yes, I think so. We'll have a little more time for questions and answers towards the end.
Thank you very much.
Okay, and thank you. And maybe Sahil, you can also tell us a little bit more about your background and the research at University of Nebraska Medical Center, as well as the program and how the program will be structured.
Sure. Thank you, Elia, and thank you, Despina, for carrying the lecture so good. So hello, everyone. My name is Sahil, and I work with Pine as a special projects coordinator, and my background is into bioinformatics. I did my master's into biomedical informatics from University of Nebraska, Omaha, and I would be continuing with my PhD in bioinformatics from University of Nebraska Medical Center, and UNMC, that's what University of Nebraska Medical Center is primarily involved with the cancer research that has been going on over there. And if I talk about my role in the whole program and Pine is to make sure that the operations are going smoothly before the program, if there are any technical difficulties, or if the users are having problems and not able to cope up with the lecture or other things. So I could help in streamlining the process. So yeah, that's me. And now I would like to show you about our resources that we have, and how exactly the program would be carried off, and what are the features we do have to offer. Okay, so I'm gonna share my screen now. Please let me know if you all can see my screen and if I am audible. Okay, so starting with our first resource, that's the educational website, or the education portal that we have, and the URL is edu.t-bio.info. We do have free courses and free projects that people are allowed um, to register and enroll themselves. We do have some of our paid courses. Those are involved more into the on the technical side, but all our basic courses are free. So you are more than welcome to explore the edu site, look around with the courses. We do have a vast collection that starts from introduction to bioinformatics, that would walk you through each and every step that is involved into the world of bioinformatics, the keywords, the terminologies, and then you move forward to more technical courses, in which we have the process of carrying the RNA-seq analysis, which would be covered in transcriptomics 1, 2, and 3. Genomics, we have the whole module that is dedicated to genomics, metagenomics, and epigenetics. You can view the course collections over here, and you're going to have all the courses that are there on our website. So these are some of the things that I wanted to mention about the courses. Over here, if I go under the project, so we do have a couple of projects that are being up, and it's available for the users to take. Most of our projects are from the publicly available data set. So we do provide you with the data set in order to play around with the data set and understand uh, more better, like how the analysis was carried out, um, and the step-by-step analysis you need to follow to come to a point where the author has mentioned his analysis or his results. So that's how the projects are designed over here. um, and I would like to tell you more about the organization page. So what are organization pages basically? So if you come down at the bottom of the edu site, you could see that there's a section that says organizations. So organizations are nothing but those are a dedicated dashboard or a dedicated group of the participants if they are a part of any of the program. So let's say that we are talking about the genomics training program. So we have a dedicated dashboard or a group for the genomics training. So if you click over here, so that's where you're gonna be coming to. So over here you could see that we have different tabs, including forum, progress, what's the curriculum, and the assignments. So you can directly go over to this organization page if you would like to register for the program. um, Yes, yeah. So over here you could see that this is the payment portal through which you can be the part of the program and register yourself. Once you are registered to the program, so you would be having the access to this organization page, and you could explore all the events. You could look at the curriculum, what all the updates are going on. You could figure out over here in the update section. uh, so I'll just walk you through on how to go ahead and pay or register. So over here you can see that there is an option that says select. So we're going to click on that, it would give me an overview about the program, what all courses are involved over here, and then you have the option to enter your credit card or debit card details. Now, one thing that I would like to mention to all the participants who are the part of the webinar today, since you guys had a chance to be the part of this webinar and are interested to know more about the genomics, so we would like to offer you a 25% discount that would be valid till Sunday of this week. So if you are interested to enroll yourself into the program, we would be emailing you with the discount coupon that you could apply while checking out and registering for the genomics program. But just to show you how exactly it works, so you can enter the card number over here, and then over here it says discount code. So whatever the discount code we are going to provide you, you can enter that. Let's say for an example, I'm going to enter Pine Bio over here, and you can click on it, you can click on apply and then submit and check out. So it's an easy process. You don't have to juggle around between a lot of things. It's just one click away to register for the program. And once you are registered, so I would like to show you a sample organization page, like what all functionalities do we have if you are the part of the program. So this is our another program that is going on currently, that's bioinformatics for the precision oncology. So since I am the part of this program, so I could see what all events happened in the previous weeks, what's coming up. So I could see all of those events. I could register individually for those events. So once you are the part of the program, all the events that are inside that program would be free for you. So you don't have to worry. It's just to register so that we know who is going to attend our next program. It's kind of an RSVP. And then you're gonna see all the updates over here. What have you been doing, and what is the file or the presentation that's used during the presentation. So you're gonna see all the updates under over here. We do have a forum, so all our participants are pretty active, and they do like to discuss about each and everything. So they keep on adding the topics over here, and then the participants do comment on each other's post, and they try to learn with the experiences. So as you could see that we have pretty good quantity of the forums over here. So we have all the presentation. We do share the recordings of each and every session that is being conducted. So you could find all the recordings over here. So that's something about the forum. And then you can have the curriculum. So curriculum would give you an idea, like, you know, what all you have to be prepared for and what's coming up. So we did start from introduction to precision oncology, then we went to introduction to big data bioinformatics. So in that way, you can keep yourself motivated to see what's coming up and at what time or at what date it's coming up. So that's the idea behind the curriculum. And you can have an interaction with the faculties directly. You can ping them or you can chat with us. We do have a chat box functionality over here. So if at all, like, you know, if you're registering to the program, or if you are going through our free courses or free projects, and if you come across with any roadblock or any hurdle, you can ping us over here, and we are pretty active in replying back. So feel free to chat with us or email us. Now moving forward, I would like to highlight some of our profiles. Those are the star profiles, and those are our previous participants who have been participating in our programs. And we would like to see your profiles as well over here in our star boards. But just to see some of the profiles, I would like to start with Ian. That's the profile, and you could see that he has a profile picture over here. He has like 2100 points. So how do you gain those points? So what's the logic behind the point? So each and every activity that you do on the educational portal, that's edu.t-bio.info, you will be assigned a point. So let's say that, you know, you registered for a course, so you're gonna get 400 points. If you complete the course, you're going to get 500 points. So for each and every activity that you're going to conduct on edu.t-bio.info, you're going to get some points. And even if you do some practical exercises on the TBioInfo platform, you're going to get the points as well. So that's how the points get accumulated over here, and you could see everything under profile. So we do have different levels, beginner, intermediate, and expert, depending upon the activities that you do and how much points you have gathered at the end. And every participant does get a badge on the profile to whatever program he or she belongs to. So if you are the part of the genomics program, you would be getting an individual badge that would say omics logic genomics program. So in the profile, we could see that he has been running quite a number of pipelines. In terms of pipeline, it means the practical exercises that he has been doing on the t-bio.info. So you could have a look over here. We do have Chutubrata Mal from MIT Kolkata, and he has been a part of the program that has been going on with MIT University, and you could see that he has around 14,000 points and he's an expert level. The other participant I would like to mention is Woki. She's from Nigeria, and she has been part of our previous omicsology program, and you could see that she has around 12,000 points. She's an expert, and then she has her social media account linked to the profile, so that all the participants can know with whom they are trying to do the program, and they can have a more clear understanding about the participants and can know each other in a better way. So the idea is to build a close community of all the people, so that they could learn, they could explore the opportunities, and they could move forward in the field of bioinformatics. Now, without wasting a lot of time, I would like to cover the last aspect. So what are some special features that are involved whenever you go ahead and take the courses? So let's say we are talking about the genomics. So over here under the courses, if you see that there's an option that says Genomics 1. So if you're going to click on Genomics 1, you're gonna see that this is the outline of the course, and over here you could see the overview, like what all things are being covered in the genomics course that's upcoming. What's the curriculum? So we do have lessons, we do have the videos, and we do have the quizzes. If you want to know more about the instructor, we have the details about the instructor over here, and the reviews of all our previous users who have been the part of the genomics program. So with the overview over here, I would like to highlight certain things. So let's say that you're going through the course and you're not very sure about, let's say, sequencing, like what exactly is sequencing. So you could hover around this specific word, and there would be a definition that would come across. So you could see that what exactly is next generation sequencing, and some of the key points that it's essential to understand sequencing. So the idea is that once you are into the course, you can be on this website, and we try to educate you at each and every level, just so that your learning experience becomes easier, and you don't have to switch the windows in order to understand the definition and then come back again to continue with the courses. So if you click on this particular lesson, so that's where you're gonna go inside the lesson, and you could see what all is being covered over here. So this is our first lesson, that says lesson 1.1. What is genomics? So you can go through it, what exactly it is. There's a video to have a better understanding about what has been covered in the field of genomics, and how could you benefit if you have the knowledge of genomic analysis. And we do have the keywords over here that are highlighted with a specific definition, which might benefit you if you're going through the courses. We do have the quizzes, just so that we do check the knowledge at each and every level, and just to make sure that the students are the users are able to understand what all has been covered in the previous lecture. So we do have the quizzes over here, and we do have the supplementary materials, so that if you would like to dig deeper into genomics and understand some of the key concepts, you can go ahead and refer to the publications. So that is about the educational website. The last thing that I would just quickly give you an overview is about the TBioInfo Platform, and that's our second resource where all our practical assignments are being carried off. And the URL is server.t-bio.info. So this is our platform where we would be analyzing our data. So we do have the pipelines, and the pipelines are nothing but those are the set of algorithms. So you're going to learn what algorithm is what and what's the use of that specific algorithm, which algorithm is required if you want to do your pre-processing, which algorithm is required if you want to map it on a transcriptome or map it on a genome, how are you going to move forward, find a mutation, and things like that. So we do have different sections over here: NGS data, mass spec, structural biology, and machine learning, which includes supervised and unsupervised analysis. And if you want to know more about these sections, you can just hover around and you could get a more clear idea about what exactly each section covers. We do have the demo pipelines. So demo pipelines are um, the way to learn and explore and play around the TBioInfo platform, so you have a better understanding of how to handle the data and do the analysis using the platform. So yeah, so that was an introduction about our resources. If you have any questions, please feel free to unmute yourself or write it down on the chat box.
Thank you, Sahil. And again, we want to make sure that it's clear how the how the program will run. So starting as you can see on the presentation that was shared, we will keep an outline of the topics that will be covered, and we will also take a look at the different online resources that support the lectures that will be carried out in a similar way like what we are doing now. So essentially we will have meetings like this. Despina, Sahil, Pratim, myself, we will be speaking about different topics. Then we will have the opportunity to do some hands-on assignments, and those hands-on assignments are going to be related to analysis of a data set that we first provide. But eventually, as you get familiar with the methods, get familiar with the data types and what can be done with those data types, you'll be able to work with your own data. And a lot of the methods that we are describing here, the purpose of them being a part of the platform is for several reasons. First of all, is so that we can follow everybody's progress and address any roadblocks that might arise as you go through the analysis. The second one is really an efficient and fast way to compare many different types of analyses without having to set up any additional resources or libraries, or wait for your own resource computationally for some of the analyses, and some of the data sets are big, so they require a lot of time. So what we want to do is standardize that process for everybody, so irrelevant of your background, everybody can do the same tasks. Now, if you're more on the computational side, all the methods are going to be described, so you're free to take those methods and build your own pipelines in your own environment on Bioconductor or other resources and put them together in the way that you see fit to address your problem. Our goal of the program is to build the logic of the analysis, provide several examples, and help everybody get through some of the major steps that are involved in such a process. Now, we hope that as a result, you will also be able to generate a new question and have your own project designed and ready for analysis, and maybe even do a significant portion of that analysis during the course of the program. The program is going to last a whole 3 months, and hopefully will be enough time for everybody to get to some level of completion in those projects. I also want to mention that the program that we have right now is one of 4 programs that we run throughout the course of the year that includes Introduction to Bioinformatics, Transcriptomics, Genomics, Epigenomics, and Metagenomics. So we do cover a lot of these different topics as programs, and then all of them also have an online component, and a lot of times programs that are being run at different universities in your area. Perhaps if you're in one of the areas that we have a program, or you're wondering about a program, you can reach out to us and we can also guide you to those programs. So I hope that this overview gave enough information for all of you to understand the program, understand how it works, understand the topics that would be covered. If you're interested to learn more about the topics and they do look relevant, you're welcome to browse online. Some of the materials that we have there can show you some of the free resources if you want to do it on your own time. If you want to join the program, we're excited about launching this next week. If you want to look at some of the projects, you can do that as well. And as a part of the program, you'll get access to this environment that we described with online learning and the online platform for data analysis that can help you deal with a lot of data. So at this point, I would just like to hand it over to a Q&A session. If you have any questions, you're welcome to unmute yourself and speak, and we'll listen and try to answer that. Or if you would like to post your question in the chat box, we're all able to address any of the questions there as well. I do want to mention that together with me, Sahil, Despina, and also Pratim are available to discuss any questions, and we'll have time to go over the 12 o'clock limit that we have. But I thank you for joining us today and for taking the time to listen to what we have prepared. Please go ahead with your questions.
Yeah, yeah. So hi everybody, this is Pratim. I will be one of the persons who will be kind of giving you some views and understanding about the various aspects of the genomics program here we are going to conduct here for the next 3 months. So it's a myriad discipline, as Despina has correctly pointed out, that there's so many things, and also in the beginning Elia has told you about the kind of things that we're going to cover. And we have an equivalent mixture of theoretical understanding and also a lot of doing things by your own hand using the T-bio platform. This is a fantastic platform which will help you to understand how actually the data analysis takes place in the real world cases, the problems that are typically answered, the problems that are typically addressed. And as you all know, that due to the advent of high throughput sequencing and high throughput data generation practices, there's a huge amount of data available, which goes hand in hand with all those in vitro and in vivo experiments. And the time is kind of correct to say that time for making those in silico practices using the computation to analyze the data and come up to a situation where you can predict. So it's a needle in a haystack situation. I mean, you have a big amount of data, you kind of precisely try to identify using the statistical methods and algorithms to pinpoint those particular area which require deeper understanding and deeper views. As of me, I am a PhD in biophysics, molecular biology, and genetics. And I've been working in the industry for quite an amount of time. And I have also taught in universities as a part of biotechnology programs in India. And I have been a part of Pine Biotech for the last 3 months. And it has been a very good journey with them, trying to understand, getting accustomed with the various operations. And I would be happy to answer many of your questions. You can get back to me via emails, which will be provided to you when the sessions, recordings, etc. will be shared. And please be active on the forum. We are trying to build a forum out here where common interested people are there. Maybe in future, some of you will be collaborating with each other to do some quantifiable and qualitative research among yourselves. So happy learning. Thank you very much.
Thank you, Pratim. So just a little more in detail about the questions about machine learning. The main point is to demonstrate how different types of machine learning methods could be applied to the data and how to overcome some of the issues because the data is very specific. So we'll talk a lot more about the different types of methods, how to combine methods, how to use them for identification of patterns, and how to use those patterns to classify or discriminate between samples and develop essentially signatures of mutations. Despina, would you like to add anything about that?
No, you covered me. We will try to give you an idea of what is machine learning and how we can use it for genomics data, because it is really difficult to use machine learning for genomics data. It's these data are very special, and no, that's all.
Okay, thank you. And as we wrap up, I just do want to mention that the cost of the program is $135, but you will get a discount code after this webinar for those that participated. So if you would like to register until Monday next week. And if there are any issues, you can talk directly to Sahil. I will just add here, the email is sahil@pine.bio. So if you have any questions about the payment or the process, please reach out to him. Also, what you see on the screen is we do provide a certificate jointly together with the Tower Bioinformatics Research Center. If you have participated in the sessions and you've actually done the things that are already there, we do have limited number of spots in the program, so please let us know if you're interested. We are going to be talking more about the program structure and the topics again in the first session that will be held on the 23rd. So unless you have any more questions, I want to again give an overview here. So July 23rd to September 28th. If you cannot, yes, we do have a calendar. In fact, if you go to this link right here, and you can take a look at the curriculum. So you will see all of the dates are right here. Those dates correspond with a week. So some weeks will have 2 meetings, some weeks will have 1 meeting, but you will see here the description of the topics and what exactly will be covered during that week. If you need more details about the times, so there will be on a consistent time, and we will provide a recording of that session. If you're not able to join, you'll have the access to the recording. And apart from that, we also hold Q&A sessions. If you have any particular subject that you're interested in, we didn't cover it, or we didn't cover it sufficiently, we will have dedicated Q&A sessions for that. All right, great. Well, thank you, everyone. Again, we'll be sending you an email shortly after this session, and we'll be starting next week. So we're really excited about starting this and hope to see you there.